Dear colleagues and friends, it gives me a great pleasure as a fellow Dutch geneticist to give the laudatio for the ESHG Award Lecture 2019 prize that has been formerly called the Mauro Bashirotto Award. It's called annual and given in recognition of individual achievements in human genetics, and it's this year awarded to Cisca Wijmenga. Cisca is a truly inspirational colleague with a remarkable career. It wasn't immediately clear at the start of her life that she would come to such height. She actually suffered from asthma and was unable to go directly to university. She first studied to be a technician, but then later on moved, fortunately for us all, to university in Groningen, after which she started a PhD in Leiden, supervised by Rune Frans, and with her promoter, actually a 2011 ESHG Award winner, Professor Gert-Jan van Omme, who is here sitting on the first row. Very proud, of course. She worked on FSHD, cloned and mapped the gene, and got her PhD awarded with distinction, which is very special in the Netherlands. After that, she moved to the States for a postdoc, and not with everybody, it was with Francis Collins. There she went into the field of complex genetics, which she continued to do when she moved to Utrecht, where she set up her own lab and became a professor and later head of department. In 2007, she moved back to the University of Groningen, where she started her studies, and where she became the head of the Department of Medical Genetics. And in 2017, she was awarded a prize which in the ESHG we also know very well, the Lodewijk Zandkuil Chair, personal chair. Her research is focused very much on complex disease, using inflammatory bowel disease and type 2 diabetes as model diseases. She combines functional genomics, microbiome studies, and organs on a chip to really show remarkable progress, and she will clearly describe that in her talk. She supervised more than 40 PhD students and received multiple awards, including the ERC Advanced Grant and the Spinoza Prize, which we would call the Dutch Nobel Prize. She's a member of the Royal Netherlands Academy of Science, and just recently she took up a new job because she continues to be very active. And 3 days ago she was officially inaugurated as Rector Magnificus of the University of Groningen. And that means something like a vice-chancellor in the UK. And this is the first female to be inaugurated at this position in 405 years of the University of Groningen. So she's really changing the field. She's very inspirational in her science that she's going to talk about now, but for many more things. And I want you all to join me in acknowledging her again for the ESHG Award for this year.
Well, thank you so much, dear ladies and gentlemen. Thanks to the the European Society of Human Genetics. This is a true honor, especially when I look at all the predecessors that have won this prize, and it's great to see a couple of them in the front row. But as Joris also said, I think it's also very timely, because when I heard about this prize last year, I could not have foreseen what a change my life would take. And as Joris just reminded you, I am taking up the position of Rector Magnificus of the University of Groningen. which is also my alma mater. So I think it is very appropriate now, because this really does not end my academic life, but it really ends my life as a very active scientist, to get the award now and end with a presentation where I show you a little bit of the insights of the work I'm doing for the last 10 years or so at the University of Groningen. And I'm really proud of my new position. It's a truly inspirational university. It's extremely old, but it's also very broad and still alive and kicking. And one of the societal themes of the university is healthy aging. And I think the university also shows that it's still very healthy. So just to show you a couple of pictures of what happened last Friday, and that was the reason I only arrived here on Sunday. And it is very traditional, where I was really getting this chain, which is still here, worn by the current rector, and I got it after the end of the ceremony. So, but if I now look back at my life as a scientist for the last 30 years or so, I started my career in Leiden as a PhD student with Gert-Jan van Ommen. I really have moved from the Mendelian to the complex disease genetics, but also more recently from human to bacterial genomes. And I really like to take up new challenges and to move my research field into new directions, as hopefully will also become clear from my presentation. But really the key to success is really the teamwork. And this is a picture that was only taken last week. And these are all the people in the research group in the department. So it's not only my group, it's everybody doing research, and it includes my own team. But it's really this collaborative effort that helps us to move forward. And that was also something very nicely acknowledged by Craig Venter in his presentation. So when I did my PhD, I was really making use of genetics to understand the disease, with the hope eventually to be able to diagnose it, to understand much better what the disease mechanism is, and hopefully also eventually to treat it. And the disease I was working on is facioscapulohumeral muscular dystrophy. We were quite lucky. At that time, it was really hard to identify disease genes, but we were able to map the gene to 4q35 by a technology called RFLPs and microsatellite markers, and I bet most of the young people here in this room have no idea what I'm talking about. And we were also able to identify the mutation, since patients really had a de novo mutation, which is really the cause of the disease. So we now can use this information to really diagnose patients. We start to understand a little bit on what the disease mechanism is, but despite that this was happening 25 years ago, we still don't have a treatment. And that also shows us how slow those processes can be. And then when I took up my first faculty position, when I came back from my postdoctoral fellowship in the US, I started to work on complex disease genetics, because I thought, well, this is really very fascinating that we now can really go to the genetics of those complex diseases, in particular because this is a major threat for human health. And I started to work on a disease called celiac disease, which is gluten intolerance. And there, a couple of years later, although I came back in 1996, we were lucky to start with GWAS studies. And by now, we have mapped about 50% of the genetics of celiac disease. We have shown that the genetics of this disorder overlaps a lot with other autoimmune diseases, in particular type 1 diabetes, rheumatoid arthritis, and Sjögren's syndrome. And we start to understand a little bit what happens in the disease. It's a disease that manifests itself in the gut, and we now understand that there is something wrong with the epithelial barrier, by which dietary gluten probably can pass through much easier, and that there is a prominent role for different immune cell types that really start off an unwanted immune response. So despite the fact that we know quite a lot of the genetics, we still cannot use this information to diagnose patients. We partly start to understand what's going on in the disease itself, that we have this overactive immune system. And although the people can be treated by a gluten-free diet, We hope, of course, that there will be other alternatives in future. And fortunately, out of the GWAS studies, we now have 16 druggable targets. So maybe there is also hope for celiac patients in future, and given the overlap with other autoimmune diseases, this information might also be beneficial for other groups of patients. But it has, and it was very nicely introduced by Greg Venter, we now also realize more and more that we also carry a second genome inside our gut, and that's our gut microbiome. And it's really due to the pioneering work of Greg Venter that we now really have a much better idea of what really is living in our gut. So this really started off with a Dutch microscopist, Antonie van Leeuwenhoek, who was the first one to invent the microscope. And really with that microscope, we were able to see and describe bacteria for the first time. And then it took a couple of 100 years before people were starting to be able to culture bacteria, although very limited, but we got a little bit of a better picture of what was going on. But now with the DNA information and the DNA technology, we can not only describe who is there, but also what they are doing. And this was already nicely introduced, and I think it's remarkable if you look at the first genome of a bacteria was only sequenced in 1995, so it's not that long ago, followed by, of course, the human genome, and then really the pioneering work of Greg Venter that showed that you can now simultaneously sequence very complex ecosystems and then recover all the individual bacteria. And this was indeed initially done on seawater, but now can also be done on the bacteria that live in our gut, and that can be made visible by taking a sample of our fecal material. So the sequencing is not limited anymore to the human genome. And with this metagenomics, what we can really now do is get a much more better picture of what is really there. So we not only describe who is there, so we can look at taxonomy, but we can, because we sequence everything, we can go back, we can really go to the SNP variation as well, so we can identify different strains. We can also look at bacterial resistant genes, and this is also a hot topic, of course. And because we sequence everything, we can also kind of deduce the function of the bacteria. And that is, of course, very important, because it's not only interesting to know who is there, but it's much more interesting to know what they are doing. So back in 1961, there was this artist, Piero Manzoni, who canned 30 grams of what he called his own shit in a can. And he made 9 different cans. And really the idea was that he had something that was really unique to the artist. And at that time, he didn't realize probably how right he was. But now we know that within our gut, there's like 40 trillion bacteria living. But now with the DNA sequencing technology, we also can learn that there is like 500 to 1,000 different kinds. And as we're sitting here, every one of us is rather unique for the bacteria we carry. And also here, like in many other occasions, bacterial diversity is really essential. And in general, the more diverse your microbiome is, the more healthy you are. So why are those bacteria so important? Well, they are really helpful and necessary to digest part of our food, and in particular fibers. But they also make essential vitamins that we cannot make ourselves, and this includes vitamin K and vitamin B. They also train the immune system, they signal to the nervous vagus, and they're also very important to our host energy metabolism. So with our microbes, we are really in very bad shape. But how can we understand and unravel much better how this interaction is between the host and the gut microbiome? And I think biobanks are really important in that. And we are quite fortunate in the north of the Netherlands, where we have a large biobank, and that is called LifeLines. So this biobank includes 167,000 participants, and that's about 10% of the population living in the north of the Netherlands. And we have been following those people since 2006. We follow them regularly. So it's a prospective cohort of all participants. And we have a lot of information on those people. And that includes data on their health, their diet, their medication, socioeconomic status, and many other things you can think of. We do a lot of measurements if they come to our clinics. So we measure lung function, heart function, blood pressure, and so on. We have DNA profiles soon of at least half of the biobank. And because it's a family base, so we have 3 generations, we hope to impute also some of the missing people that we are not able to genotype. And then we also collect every 5 years biomaterials, initially urine, plasma, and hair, but now recently also fecal material. And with that in hand, we started out a kind of proof of concept study back in 2014 to better understand what are really the factors that drive the diversity and the composition of the gut microbiome. And since all the data in this biobank is collected in a very standardized way, we were able to really correlate the metagenomics data with different dietary factors. So we had 78 in total, 44 categories of drugs, 5 categories of smoking, 39 diseases, because keep in mind, this is a cross-section of the general population, so we have people that are sick as well, and then 41 intrinsic factors. So this includes sex and age, but also blood lipids and so on. And by doing so, we found factors that are kind of good for your microbiome, and if I say good, I mean they really increase the diversity. And these are food items that we all know are kind of healthy as well, like fruits and vegetables, but also red wine, and that might be not necessarily on your top list of the good food to eat or drink. And then there is bacteria that are kind of bad in the sense that they reduce diversity, and this includes a lot of the processed food, but also being obese and drinking beer. And then there are factors that are really, really ugly. And with that, I mean that they change your microbiome so much that they really increase your risk for very nasty infections that can even be life-threatening. And on top of that is really the proton pump inhibitors. This is medication that people use to treat heartburn. And the problem really with the PPIs is that people use this for many, many years. And we show that you change about 20% of your taxa if you, for a prolonged period of time, take the PPI. So this is something that is now being discussed in the Netherlands, where this drug can be taken over the counter if we should really change that and only prescribe it by a medical doctor. So, but if you put all this together, so we checked about 200 factors, we really saw that 126, so more than 50% of those, could explain nearly 20% of the variation in the gut microbiome composition. And that is quite a lot if you think in terms of genetics, where we usually can explain only a few percent, in particular if you keep in mind that we were only looking at 1,135 individuals. And these are a combination of all the exogenous and the endogenous factors we looked at. So we can also really conclude from that that the role of the environment really seems to dominate over the role of the host genome. And this was also followed up in a very beautiful study that we conducted in collaboration with a group of Iran Segal from the Weizmann Institute in Israel. So people have been doing the genetics, so here at the genetics conference, have been looking at the genetics of the microbiome, which in itself is of course a very complex trait. And work from Tim Spector in twins also showed that about 10% of the bacterial taxa are heritable, and that means that you only can look at those to find also genetic host variants that really associate with bacterial taxa. But we also should keep in mind that this study are really early days, because all the studies are really small if we compare that to all the other studies we do on quantitative traits, and where nowadays people are doing tens to 100,000s of samples. So these are the only studies being published so far. There's 3 studies from the UK, Canada, and Germany where people make use of 16S rRNA sequencing, so they only look at the 16S gene, and then our metagenomics sequencing study. And if you compare the results from those, you hardly see any overlap. And I really think that is because the studies are underpowered. So to overcome that, Sasha Zernakova from our department, together with people all around the globe, started the MyBiogen consortium to really do a big meta-analysis. So they now have compiled more than 15,000 samples. They come from different ancestries, and they're all based on 16S rRNA data, since this is still what most of the studies have been using. And these are just pictures from the very preliminary data, where we either treat the microbiome as a binary trait, so either it's the absence or the presence of a certain bacteria, and then we only see 12 genome-wide hits, or we treat it as a quantitative trait, and then we see 28 genetic loci that really affect the abundance of different bacteria taxa. And you notice, of course, there's a very strong signal over here on chromosome 2, and this is the LCT locus, the lactase locus, which really affects the abundance of bifidobacteria. And we had seen this before as well in our LifeLines cohort study, that if you take the people that are lactose tolerant, so they're either the homozygous and the heterozygous carriers, they have lower levels of bifidobacterium compared to those that are lactose intolerant, which have higher levels of bifidobacterium. So this was all known. It's a recessive trait, but we also have shown in the past that this only happens in people that drink milk. So again, we partition here the data in the lactose tolerant ones and the intolerant, which is the red line. And if you do not drink milk, the level of bifidobacterium is exactly the same. However, if you do drink milk, and you would think that people that are lactose intolerant do not drink milk, but they do, to the same extent as people that are lactose tolerant, but they have much higher levels of bifidobacterium. So they really compensate for the fact that they cannot, for the lack of lactose activity, and that's compensated by the bifidobacterium. So this is really how nature deals with those. But now, with the data from the MyBiogen consortium, we also see another interesting effect, and that is that the effect is much stronger in adults, and that also the associations are really population-specific. Since we now have different ethnicities, we also see its absence in some of the populations we study. So this shows us how complex it is to look at the genetics of the microbiome, and probably all those factors, the influence of diet, but also the age of the population and the location where you live, which probably says also something about your local diet, can really dilute the signal. And I'm confident this is also true for many of the other loci. It might be one of the reasons that we do not pick up so many signals. So since we now have a much bigger cohort that we study, so we started the LifeLines 10K cohort project, so we did a little bit over 1,000 samples before. We now have sampled from 10,000 people bacteria from 3 different body sites, being the nose, the throat, and again, stool. And in such a way that we not only isolate DNA for sequencing, but we also have captured material in such a way that we can go back and culture live bacteria. So we just finalized the metagenomic sequencing of the 10,000 people, and we also geomapped the position where the donors are coming from, and are nicely spread over the 3 northern provinces. But what is also nice is that some of them really live in the big cities, like Groningen or Leeuwarden, and some of them live in really rural areas, which also allows us to really see what is the effect of that. And we really see differences. So depending whether you are from an urban or a rural area, you may have lower or higher diversity of certain bacteria. And this is just one example where we see this for this Cittarella species, which is much more abundant in people living in cities than in the rural areas. And this is a bacteria that was originally isolated from canine feces. So we do not know if this is because there is there's more people with dogs in cities. It's something we have to, of course, figure out. But it's also one of the bacteria that's also associated with inflammatory bowel disease. And there's many more of those examples. But we also saw that the gut bacterial abundance, and this was known, of course, from the twin studies as well, is also affected by genetics, but also by cohabitation, so people living together in one house. So this is one example of a bacteria that's under more genetic control. So here we compare people that are related to each other, so being sibling and child and parent pairs, and they have more similar microbiome, where's my mouse, than partners, whereas the partners live in the same household, and the partners are very similar to any random pairs. On the contrast, we also see bacteria like this Provatella species, where if you live together, you have a much more similar microbiome than any random pair. And this is important, of course, because this is something we also now can take into consideration in our analyses. But I think it's also important to think, well, how can we now use the microbiome in healthcare? And I'll give you 3 examples how we think this can happen. I need some water. So of all the studies that have been published so far, they're all correlations. So they correlate bacterial abundance and composition with the presence of a disease. But the question always is, of course, is this cause or consequence? So do you have the disease because you have a certain microbiome, or do you have a certain microbiome because you have a disease? So Serena Sanna in my group, in collaboration with Mark McCarthy at the University of Oxford, set out a proof of concept Mendelian randomization study, where, like in a clinical trial where you test the effect of a placebo and a drug on the outcome, namely being a difference in the prevalence of a disease, we also can take genetic instruments to really see if different microbial compositions also have a different effect on a phenotype. So in this case, we were looking at metabolic traits, including type 2 diabetes. So we could make use of the fact that from all our LifeLines participants, we have host genetics, which we can correlate to the gut microbiome composition. Again, this only holds for those that are heritable. And we also had fecal short-chain fatty acids. So these are the fermentation products made by bacteria from fibers. And that we can also correlate to host genetics and then use the genetics instruments that are linked to those features to the metabolic traits. And if we do so, we indeed were able to observe that SNPs that are linked to either short-chain fatty acid levels, to certain bacterial taxa, or to certain pathways are causally linked to different metabolic phenotypes, including type 2 diabetes. But if we do the reverse, we take now SNPs linked to metabolic traits, they are not associated to the bacterial composition. So really showing that the microbiome is causing the phenotype. And we also were able to show that this is also associated with changing levels of short-chain fatty acids, in particular butyrate and propionate. So we saw that if you have increased levels of butyrate produced by your bacteria, that is good. So if you take the genetic predictors of the butyrate-producing pathway, we saw that you have a much better insulin response, and this is something that's good because you can deal with your sugars much better and quicker. And this was all related to well-known butyrate-producing bacteria that correlate with this pathway, in particular Eubacterium rectale and Rosabacteria intestinalis. So you have to keep your butyrate production high, but at the same time, you have to keep your propionate levels low, because if you take now again the genetic predictors of fecal propionate levels, then we see you increase your risk for type 2 diabetes. So showing that this balance is also very important. But it is very good news because now we know of this direction. We can in future think of other ways maybe to treat type 2 diabetes. On the one hand, maybe we know now how we can change our diet in such a way that it directs our microbiome in such a direction that it's more beneficial for our host metabolism, or we can maybe also develop new classes of drugs based on our microbiome. Well, that is work that still needs to be done, but I think this is a very promising direction. And Hippocrates, the father of medicine, already knew that, of course. He stated, let food be the medicine and medicine be the food. All disease begins in the gut. But another way where you can think of how you can use the microbiome is really as a biomarker to diagnose diseases. And I won't go into this example. It's also published. It's work by 2 talented PhD students in the group in collaboration with Rins en Wiersma, also from the UMCG, where we were able to show that you can really use a stool to really distinguish irritable bowel syndrome from inflammatory bowel disease like Crohn's disease and ulcerative colitis. So from the outside, the diseases look very similar to each other. They're characterized by diarrhea and also all sorts of complaints. And the only way to diagnose now is to do a very invasive endoscopy. But then if you go into the gut of an IBS patient, you see it's completely normal. Whereas in the IBD cases, you see all kinds of ulcers. But we have shown now that you can take a little bit of fecal materia and really distinguish those two. So this is something we now have to test in a prospective study to see if the positive predictive value is as good as what we predicted in our cohorts that we have been studying so far. But again, I think this is very promising. And then a third direction, which I find extremely exciting, is that it also has shown now, and this is not work by us, that gut bacteria can really enhance the therapeutic efficacy of medication. And the best example is the example of immunotherapy, where we see lots of great stories coming out now. But it's also clear that not everybody with cancer will respond in the same way to immunotherapy. And it has been shown by a couple of very beautiful studies last year that depending on the gut microbiome, you do or you do not respond to immunotherapy. And there they did studies where they first treated people with antibiotics to change their microbiome to really get a better efficacy. So I think this is also an area where we'll see a lot more developments, that we can also make use of this type of information to hopefully treat people much better. And this therapeutic efficacy is, of course, not only true for immunotherapy, but for many other drugs as well. But yeah, if you think that using poop bacteria for medicine is something new, it isn't. It was already described in the 4th century by this Chinese medical doctor, Gi Hong. And what he figured out is that if he gave yellow soup, and I leave it to your imagination what yellow soup might be, that he could really treat people with all kinds of different phenotypes. And nowadays we call this fecal microbiota transplantation. So I think it's always good to look back in history to see what people came up in old days. But yeah, having all this information, we still really do not understand how this all works. So how do the bacteria signal to the host? So how can we really move to a better mechanistic understanding where we can incorporate the microbiome, the short-chain fatty acids that are produced by them, the epithelial barrier, because this is, of course, really the first contact of the microbiome with the host, and also our immune cells that play a very important role, in particular in the gut. So we decided 2 years ago to really move our attention to tissue chips, to really model disease, and this would also be a way to develop drugs. And this was also featured earlier this year by National Geographic as the future of medicine. And this is really how the chips look like. They are transparent, so that means you can even put a microscope on top of it if you really want to see what's going on. And the beauty of it is that you really can recapitulate tissue and organ level physiology. In our case, we would be interested in the gut epithelial barrier, but you can, if you have your IPSC cells, think of every cell type you're interested in. You can add the microbiome or short-chain fatty acids, as well as the immune system. And our starting point would really be induced pluripotent stem cells for the reason that we can then decide what cell type we want to make afterwards. And really the beauty is that you can then choose that from any desired genetic background you're interested in. And that really allows you to make very personal models of disease. And in particular, if you think about complex diseases, you can take into account this complex genetics. And now with all the polygenic risk scores, we can really select the people we're interested in. So again, we go back to our biobank, our LifeLines participants, where we can take blood, and from that we can isolate erythroblasts, and we can reprogram them, in our case in gut cells, and then see them on those chips. And they really form this phyllis-like architecture, very similar to what happens in our inside of our body. And if you really look into what cell types are present, you really find back the cells that are also normally found in the gut, including enteroendocrine cells, goblet cells, and so on. But then I just told you that we have this cohort now for which we also have stored live bacteria. So now we can also go back and, depending on what we find with our metagenomic sequencing, culture any bacteria of interest and then really seed the bacteria on top of the chip so we can really look at the interaction. So this is how it looks in real life. So we either select people on the basis of the polygenic risk score or on a disease of interest. We can also use urine, but this is not as practical as taking blood. We isolate somatic cells, so if you go to urine, it's the kidney epithelial cells, and from blood, it's the erythroblasts. You reprogram them into iPS cells, and then you can make organoids from that. And in our case, these are intestinal organoids. We dissociate the organoids and we see them on the chip. And what happens once you do that? So this is a cross-section of the chips we use. They are from Emulate in the United States. So there is a membrane here, and your cells are seated on the membrane. And then there's an upper channel indicated here in blue, which kind of represents the lumen of the intestine. And there is the bottom channel, which kind of recapitulates the blood flow. And there is also 2 vacuum chambers on each side of the chip. And by putting pressure on it, you really kind of mimic the peristaltic movements that are also present in the intestine. And that is really the reason why you get this villus architecture. And then we can use the upper channel to flow through the microbiome or the metabolites produced by the microbiome or whatever we want. And then on the bottom channel, we can flow through either whole blood, but that can be clotted, or even immune cells. And we can even grow endothelial cells to kind of mimic blood vessels. So this is really exciting, but I'm also happy that we have funding to do this for 10 years because it's also extremely complicated. So in conclusion, I think if we think about genomes in health and disease, and at this meeting we have talked a lot about the host genome, and that is of course great because it's stable. In theory, you only have to analyze it once, and preferably you would like to do that at birth, so you can use it over and over at different stages when you need it, except of course in the case of cancer, because your cancer genome is evolving all the time. But now we have to be aware that we also have our second genome. The problem, however, it's much more dynamic than our host genome. We probably have to analyze it multiple times, but you want to do it at least during periods of homeostasis, and that is pretty stable, but then also at periods of perturbation. And I think the beauty of our second genome is that if we think about treatment, it's something we can much easier probably tune to hopefully also affect our health, so we can also live a little bit, not so much longer, but more years in a healthy situation, as was nicely also shown by Craig Venter. But yeah, pushing those boundaries of science, which I really have tried to do throughout my career, really only can be done when you have a very collaborative, but also interdisciplinary environment. So these are the people in the UMCG that do all the microbiome work. So these are my heroes. We call it in-house the POOP group, but it's our microbiome team. And this is really the group of the Netherlands Organs on a Chip initiative, where we do all the cool stuff with the tissue chips. It's 6 groups throughout the Netherlands. It includes people from the technical universities, because there are lots of nanotechnology involved. There is geneticists, microbiologists, and it's all headed by a famous stem cell biologist, Christine Memory, from the University of Leiden. But of course, I should acknowledge many more people, because there are many more people involved, as well as all the funding bodies. But I really would like to end with a big thank you to many of my genetic friends and colleagues, and many of them are in this room, for the fantastic journey I had over the past 30 years. So thank you for your attention.